This is Dr. Jesse Kleingardner, here to tell you more about research in the Kleingardner Lab at Messiah College. If you are interested in joining the lab, then feel free to come chat with me or shoot me an email to set up a time to talk. My research focuses on metalloprotein engineering, and a little bit of the justification behind that is that, well, metal-containing proteins can do so much. Here are a few examples of just heme, which is a certain type of metal cofactor found in proteins, and some of the different functions that they can do. They include catabolizing, breaking down metabolites or drugs like in cytochrome P450. They include sensing nitric oxide and guanylate cyclase. They include transferring electrons in cytochrome C or catalyzing the reduction of oxygen in cytochrome C oxidase. But wait, there's more, even more functions. Heme proteins are very versatile. The research projects in my lab that focus on heme protein engineering are in order to expand the known functionality of heme proteins or tune their properties or add a new function like catalysis of a specific chemical reaction, so that we can really expand our knowledge in how to design our own custom-built heme-based proteins. A lot of them involve things like mutagenesis, switching out an amino acid of a heme protein for another one, or metal substitution, substituting the heme iron for something else. And this has been done before to expand the known functionality of heme proteins, to design a new function into it. But we are exploring that possibility through different methods of substituting metals that are more efficient, or putting them into different templates, different heme proteins. Another project involves taking a non-heme protein and actually turning it into a heme protein, inserting new hemes. In particular, this one involves stacked hemes, which would mimic a oxygen reduction metalloporphyrin catalyst if we were to combine it with metal substitution. The types of projects involving heme proteins in my lab focus on a range of different methods, starting out with cloning, making expression vectors with a gene for a protein, mutating those expression vectors with mutagenesis, putting those into E. coli and expressing the protein in E. coli cells, purifying the protein, and then characterizing the protein to see if it has the function or properties that we intended. One of the other projects involves fluorescence-based metal sensing. In particular, we're starting with this template that binds to calcium. And when it binds to calcium, it changes shape and it brings together a couple fluorescent proteins. And so what started out as a fluorescence around 475 nanometers ends up fluorescence closer to 530 nanometers when calcium is bound to it. And so this has actually been used to measure calcium concentrations inside the cell, and it's completely genetically encoded. This way you can see a lot of signaling processes happening inside of cells. Well, what if we could redesign this to bind to different metals? We can make a genetically encoded iron sensor or copper sensor. In order to do this, we need to mutate all those calcium binding amino acids to amino acids that bind to other metals and randomize them rationally. Then we can see if that calcium sensor binds to any other metals and changes fluorescence as a result. And in order to do that, we're going to use a high throughput fluorescence screening, so we can screen a number of mutants at the same time. The techniques involved in this project are mutagenesis, expressing proteins in cells, and then screening those cells for their fluorescence, and then finally analyzing it with DNA sequencing. There are a number of other projects in this lab, and they focus on, well, a really wide range of techniques, but all core techniques in biochemistry or some in physical and inorganic chemistry as well. So if you're interested, come talk to me.